Yeah. Oh, there she
It
is.
is.
Hi, Susan.
Hello? Can you hear me?
Yeah, I can hear you.
Oh, okay, that's good.
Yeah. No.
So, yeah.
Should he joined last week, so.
Yeah. Oh, welcome.
There goes the phone.
Already.
Yeah, I got my phone answer in the house.
Ah. So, Susan, you sent me a talk on the tissue timeline, I think it is.
Yes, and I could bring it up and try to share my screen if you want, but I don't think that'll work.
Well, yeah, I'll try it, and then I have it on my side if you can't.
Yeah, okay. Okay, so there it is. Click on there, and then we try to share screen, wherever that went.
Huh.
The share is grayed out.
Okay. Well, I'll share on my end, and then So let me share my screen, and you can tell me when to advance the slides. We can do that.
Sure.
Yeah. So let's see. Okay, this is tissue timelines.
Yes. So I've been struggling with this issue because I have to image tissue while perturbing it in some manner, compressing it or pulling on it. There's not that many studies on tension in tissue, but there's most of them are actually compression of tissue. But you have to be aware of what's normal for a tissue or physiologically normal and what isn't. For instance, brain tissue is quite sensitive to compression and tension, and say muscle and tendons are not as much. And look at this time scale, and you can see why number one, tissue is complicated, and number two, why you have to be careful when compressing it, so that you get some sort of meaningful measurement. So yes, then you get cell deformation, and it happens in minutes. So if you're going to do an experiment, you have to be aware of that. And a lot of our tissues are used to being compressed with our heartbeat, for instance. Like that's the natural way of that's their life. If they don't have compression, then they'll change cell, for instance, or undergo apoptosis. That's especially true in our blood vessels. The endothelial cells in our blood vessels are sensitive to flow, for instance. And if they don't have flow in their life, flow of blood or something, and pressure, then they'll revert to a different cell type, remodel. I have a lovely paper on that if you're interested in seeing it. And then I found this graph, Smith-Strong graph, there's just one paper about this. I can't find anything else, so you can see this goes from milliseconds to seconds, and you can get hypoxia due to stopping blood flow because you're compressing it, and before that you can get what's called hyperelasticity. If you hit something too hard, will cause damage. So it is between those two extremes. So you only have a little part of this graph that's in the green.
Susan?
Yeah.
Does hyperelasticity basically mean beyond the linear stress-strain relationship?
No, it's a fast moving force that's applied to tissue. Like they've actually, you want to do hyperelasticity.
Okay, then that suggests that we're dealing with viscoelasticity.
Yes and no. It's hyperelasticity. Hyperelasticity is different. Anyway, you actually want to study that if you're studying brain injury, for instance. Anyway, so you get apoptosis or necrosis depending on what's happened to your tissue if it's outside the green and yellow range. Tissue can recover from experienced hypoxia and can recover from blows, but it takes a while. So if you're trying to measure live tissue, you have to be very aware of where the tissue range is that you're dealing with, because you, like I said, you've got softer tissue that seems to be more sensitive to compression, for instance, and then you've got other tissue like muscle, and it's used to being ripped apart and shunted around and stretched, and like that's how you build muscle, is that you actually cause some damage to that muscle and then it rebuilds. So that's how you build muscle bulk.
Yeah.
Yeah. Anyway, but that doesn't work for brains. So anyway, so I don't know. Next slide there. So there's your elastic curve with the stress and strain relationship. Stress on the up and down axis and strain on the lower axis, and stress is the force per unit area and strain is the amount of change in the length of the tissue. And if you've got elastic, it's supposed to be a nice straight line, and if it's hyperelastic, it does this little lovely S-type curve.
All right, Susan, is the hyperelastic curve dependent on the velocity of impact?
Yeah.
Yes. Okay.
Yeah, it depends. Yes.
Yeah, I see what you mean. The elastic limit is when the curve stops being a straight line.
Yeah.
Okay.
Yeah.
Yeah, and that's just part of this curve. I guess I should have included the rest. I mean, if you put too much strain on something or too much stress, then it will deform permanently.
Okay, another question about this. In hyperelasticity, do you get hysteresis?
I haven't studied that much. I was trying to aim for things that weren't hyperelastic. I have a whole thesis on it if you want me to send you it. Hyperelasticity in brain. It's non-linear for sure.
Yeah, but is it hysteresis would mean if you're going to the right, the curve goes one way, and if you go to the left, the curve's different.
I suspect it has hysteresis. In other words, it has energy storage and release. That would be my guess.
Yeah, something that's elastic, that you just go back down the straight line.
Yes. something that's properly elastic. You do that.
Mhm.
Not something that's viscoelastic, but something. And tissue is to be viscoelastic on top of it. Anyway, so I have this lovely dilemma. Yeah, these are okay. These are the measurements you take when you're dealing with tissue, and it's relaxation, creep, and you're looking at whether something is thixotropic or rheopathic, and and you can also do oscillatory measurements. And you have to be careful about the frequency of the oscillatory measurements. And some people who are working in the field think that they should just simply use the heartbeat as the perturbation in their measurements, because that's what's natural.
Yeah.
Anyways, did I put in another slide here? I hope.
Yes.
Okay. Because then I've explained myself. So you have elastic properties. So there's no hysteresis. It goes up the graph linearly and back down. But you have viscoelasticity, and it'll flow like a fluid under the right conditions. Plastic, and that deforms permanently. So you end up with a graph that's viscoelastic and plastic for most things. And our relaxation time, that's when you apply a constant deformation. And then creep is when a constant load is applied. And for creep, I like to say that's where you've got your object and you've put a weight on it and leave it there. So that's like a constant load, and it will change shape with the constant load. And then if you go down here to thixotropic, what is that? It will become more fluid as force is applied.
That's the case of ketchup.
Or clay, pardon?
Ketchup is the classical example. Right.
Yeah, of thixotropic. But then you can get rheopathic, and that means it becomes stiffer as force is applied. And our synovial fluids in our joints is actually somewhat like that. So, anyways, this is, I'm having to think about all of these things so I can get a proper experiment to measure the viscoelasticity of something. So you almost need to know what you're measuring before you can decide what parameters you're going to use.
Yeah.
Or most likely, that's what I think. So if I'm going to measure an insect's sphericals, then I need to know that, and then figure out like what type of forces I should use to measure their viscoelasticity. But if I'm going to measure a mammalian brain, well, then I have to be really careful about forces applied, because even a scalpel cutting into a brain, the force of the pushing on the brain is actually a trauma.
Right.
They're very sensitive. So,
Okay, Susan, all of these concepts are assuming or not assuming anisotropy.
what?
Anisotropy, in other words, that the reaction to a force is the same in all directions.
One has to measure it in all directions because tissue is anisotropic. I also know that.
Okay.
Yeah.
Okay, that means that really we're dealing with tensors then.
Yeah, tensor fields. Mm-hmm.
Yeah.
Yeah, 3D tensor fields. Actually, it's 40, of course, because time is obviously involved in this.
Right.
Yeah.
And one has to choose if they're going to send an ultrasound wave through something, that it doesn't damage the tissue, and that it's at the right frequency so that it doesn't cause problems for the tissue. But I don't know. I've seen some burst ultrasound used for this optical coherence elastography that I'm supposed to do, and I think it's tissue damaging. So I'm not sure how to prove that. Yeah. Well, it looks damaging. You look at the waveforms.
Yeah.
Well, apply it to an embryo and see if you get developed.
Yeah, I guess that's the proof. Aim it at an axolotl egg, and if it dies, well, you know it was bad. I know that optical coherence tomography, that actual tomography, the eggs survived that. We put a couple into the system and took images of them as they were developing, and then they developed normally. And my friend Elizabeth has what she calls a scanner baby, and the axolotl actually had more babies. They were normal. So
Normal,
that
okay.
system, we know the optical coherence tomography is okay. But as to the ultrasound, first ultrasound, I'm not sure.
Yeah, you might want to check the literature on toxicity tests using embryos or eggs. There's a fair literature on toxicity testing. Zebrafish embryos some are probably more delicate than others. Could be that the axolotl is particularly tough.
Okay, well, zebrafish, that would be good. Maybe somebody's done some ultrasound or tried it with zebrafish. I don't know. Ultrasound is kind of a large wave.
Usually they're done for, what do you call them? Teratotoxins. They're looking for chemical effects, but the mechanical effects maybe needs its own field. There was some concern, for example, that ultrasound of a woman with an early pregnancy could be dangerous to the pregnancy.
Well, I thought it was dangerous just because the ultrasound seemed to warm up the tissue.
Well, okay, the reasons might be that, or could be vibrations, but in any case, there might be a small literature on that. Ted Lyons, I learned about that from Ted Lyons in radiology at U of M.
Oh, okay. Did you think there was an effect?
I think he might have been investigating it, but I've never seen the literature per se. But I don't have anything on it.
No, my main objection to ultrasound, and this is my third pregnancy, was that it warmed up the area. And I know how bad that is for an embryo. You shot things.
Now, sometimes you do it deliberately. Are you familiar with lithotripsy?
No.
Okay, lithotripsy is when you send shock waves, sound waves, focused through the body to break up things like kidney stones.
Oh, okay. Lithotripsy?
Yeah, lithotripsy, yeah.
Yeah, okay. Well, I should look that up because I swear some of the ARF or focused ultrasounds are damaging. They short bursts, tick, 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 and then nothing.
I've been through it.
Tick.
by the time you're done with the thousand sound bursts, it hurts.
Yeah. Well, I know someone who had their kidney stones removed this way, and they were not happy. They were bruised.
Yes, it's similar to being slapped on the back 1,000 times.
Okay. Well, if you're going to slap an embryo, it might not survive the experience.
Okay.
Anyway, that's my presentation, and that's my issue. Like, there's not that much literature out there. I feel like I'm swimming in the dark, and so that's a weak thing.
Stupid general question. What is the state of the art in doing three-dimensional reconstruction of tensor fields?
Ask Dr. Sharif. He's the guy that loves tensor fields and going, okay, you can have it. Yeah.
Well, invite him to give us a seminar on reconstruction of tensor fields.
Yeah. Have to look into that.
I'll ask him if there is such a thing. So if he's done any work with that.
Yeah.
And it's a general problem, obviously. Kind of independent of the physical property being measured, as long as it's got represented by a tensor field.
Well, he has done a lot of work on that type of matrix manipulation. Like he had some actual published math papers on the subject.
Okay.
That's Dr. Sharif.
I had a question about you mentioned something briefly, and I know a little bit about this area, is that you have, if you apply forces to cells or different mechanical stresses and strains, you can actually trigger cell differentiation, or you can trigger, some cancer pathway or something like that, as well as apoptosis. So, I mean, yeah, there's a literature actually on, like, people trying to induce cells to become stem cells through applying different forces to the cells, and then other types of differentiation. I don't have any references on me, but I know that people have done this kind of work.
Oh,
yeah, we had
that would help me out, because then that usually acts as a nice springboard into the literature, if you can find one.
Bradley, we had a funny example in our lab where we tried to mount an axolotl embryo on basically on something that looked like a golf. What do you call those little things that you put a golf ball on?
Oh, golf tee.
Yeah.
Golf what?
Tee.
T.
Yeah, T.
Okay, except we made it hollow so that we could put some negative pressure to hold the embryo from falling off. Okay?
Okay.
And the result was the embryo just dripped down the pipette.
The axolotl eggs, they're slippery. They do all kinds of strange things for you.
Well, no, it actually sucked it down, so part of the embryo went right down the tube.
Oh.
Yeah.
That slow negative pressure. It's an example of viscoelasticity. Constant slight pressure goes right down.
Yeah.
Oh well, I'm not supposed to be just thinking of embryos. I'm supposed to be thinking of all tissue, and I'm going, well, if I'm going to do any experiment with this, I have to know the tissue ahead of time in order to set up the experiment. And I also need to know whether you want hyperelastic forces or not.
By the way, have you looked at bone?
Yeah, people like to study bone. I've
got
seen it in my biomechanics class.
Okay, yeah, it's highly anisotropic. And there's probably all sorts of studies on breaking strength and things like that, forces you can apply to it. Yeah.
Yeah, Dr. Zhang did an experiment on chicken bone where he broke the bone sideways, and he did it like he would have done a piece of wood to see its strength. Yeah, it was an experiment we did.
Oh, you didn't do the wishbone experiment.
Not the wishbone, no. A leg bone.
I know there are people interested in looking at something called brain multiphysics. So it's like where they, usually people study the brain, they study neurons and some of the supporting tissues, but there are people who model the brain or the nervous system as like a set of different types of tissues, and they study like the forces that, are involved in development and in everyday. So, there's that. I'll have to point that out to you. There's a journal called Brain Multiphysics, and I don't know if it would be relevant here.
Oh,
but,
really?
yeah.
Well, I have a paper. It's called 50 Shades of the Brain.
Okay.
I know, but
Yeah, okay.
it's memorable, and the researcher did multiple experiments on brain tissue. And there's a definite difference between white and gray matter. And she also tried to apply shear stress. Then I took a look at the experiment and I said, okay, complicated stress. Because she compressed it and then she sheared it. And I'm going, no, that's not shear stress, that's complicated stress.
Okay, let me give you two more examples that might be important. One is in hydrocephalus. In hydrocephalus, the head becomes very large and the brain becomes quite distended. But the intelligence seems normal.
Well, people die from that.
Well, yeah, usually, and nowadays they put stents in, so you don't see people.
Yeah, the stents rescue.
The only one I ever saw was a kid in China 40 years ago.
Oh, well, I saw one in my neighborhood 60 years ago. Yeah,
Okay, yeah.
something like that.
But in any case, the point is that the cerebrospinal fluid apparently keeps increasing the pressure on the brain, but very slowly.
Yeah, and the child will become retarded unless you add the stents. And they add the stents before the child is born, actually, because it's a cesarean birth otherwise.
Okay.
And the brain is already damaged. So.
Okay. Okay, so maybe I'm wrong about it being normal.
No, not normal intelligence after a while.
After a while, okay. Okay, but it's another case of slow pressure being applied to the brain.
Yeah.
Okay, another case, which is more down at the minutes level, is breast compression during mammography.
Yeah. Did you know those machines were designed by a mechanical engineer? I don't know, but I betcha.
Been there, done that, huh?
Yeah.
Okay, well.
Written all over it.
Okay, what's the effect of breast compression on the tumors they're looking for? Or on the normal tissues?
Seriously bothers them, probably.
Okay.
Does it to grow more, perhaps?
Yeah, I'm just saying it's another case where you want to look at forces applied to tissues.
Yeah.
Yeah. There's an idea. I'll do a study on that.
Okay, good.
Yeah. Oh, yeah.
Yeah.
Okay, because Krishna and Shruti and I are starting a project which involves breast compression. And the standard for breast compression seems to be to compress the breast down to 3 to 5 centimeters. Which is probably a lot.
Yeah.
It depends on the breast. But anyways.
So be interested in what you find if you do that.
Yeah. Yeah.
Anyway, you can tell Turkey my joke.
Oh.
It was designed by a mechanical engineer.
Yeah.
Yeah.
Let's see. So yeah, thanks for that talk, Susan. And I wanted to talk a little bit about some of the stuff that Dick and I have been trading a couple emails about Bacillaria and some of the future things.
Before you start, I do have to leave at 10 because
Okay.
I have to drive to Winnipeg. So,
All right. That's fine. Yeah.
so you know.
Okay.
Okay. Good enough.
Yeah.
Okay, have a good day.
Yeah. So let's see. It's here, this acceleration analysis. So this is, Dick sent me this acceleration noise biography. This is on acceleration noise. So we're measuring, last week I was showing the measurement of velocity and acceleration in a single Bacillaria colony. And he sent me all these references on acceleration noise and Gear, and it looks like they've done a lot of work on this in traffic engineering or in vehicle engineering. Yeah, so this is good to look at, like some of the things they've been doing there, and then so
That's the only place I found it. As I said, the phrase acceleration noise is used for other things, but they didn't seem to be noise due to linear movement.
okay, so it's linear movement. Yeah.
I was concerned with linear movement because, a car with acceleration noise would seem analogous in some way to a diatom.
Right.
Okay.
Yeah. So that's, and then how to conduct this analysis. So Dick gave me some ideas of how we might do this. Plot acceleration noise under spike frequency versus position or velocity. Compare reversal velocity and acceleration to same during Bacillaria reversal. Plot velocity, pair length versus overlap of the two cells. So I've gotten made a little bit of progress on one, or something similar.
Oh, okay. Good. Good.
And then two, I looked at the data last week. I was looking at it, and I was trying to figure out. It looks like if you take the vector of velocities, it looks like the velocities, like they'll go, basically they're positive going from one end to the other, but there are some points that are negative, and I don't know if it's measurement error or if there's some variation there, but I'm trying to figure out how to characterize it. I haven't been able to get at it yet, but just looking at the data, I can tell that there are some points where there are some reversals in the velocity. So I don't know exactly. I'll have to.
No, that's not what I meant.
Oh, okay.
I didn't mean the instantaneous reversal. I meant the periodic reversal.
Okay.
Of the whole diatom.
Okay.
Okay. I mean, yes, we're looking for both, but when a Bacillaria cell in a colony stops and reverses, are its properties similar to when a single diatom stops and reverses?
Okay.
Or can they be distinguished somehow?
Okay. All right. Yeah. I'll kind of think about it in that way too.
That may be tied with point 3 because Bacillaria, again, I don't think anyone's measured this for single diatoms, but if a single diatom's moving and then suddenly reverses, how sudden is that reversal? Or is it similar in time scale to the time scale for reversal of Bacillaria?
Right.
Okay? In other words, there might be a distinction between the colonial behavior and the single cell, but we've already shown that it's not in the noise.
Okay, yeah.
Though there may be some qualitative differences in the noise. That's why number 1 is there.
All right.
Okay.
All right. Yeah. So that leads me to the different graphs that I created this week.
Okay.
So this is hard to get at here. Let's see. So this is position versus acceleration. This is the position of the cell versus the acceleration of the cell. You can see that there's some outliers here of the acceleration. And so this isn't like a time series trace. This is just the points. So you can see their outliers out here. And so one of the things I did was to get to create like a filter where I took like a threshold of about maybe 0.25, and everything above 0.25 or below negative 0.25 were considered spikes. So this is sort of spike classification for this trace.
Okay. Yeah.
And then looking at this, then you can take the acceleration curve, and then this is the acceleration curve as continuous data, but these are the spikes in this blue.
Spikes,
Yeah. Is classified by this. So,
okay.
yeah. And then we get an interval count. So the interval between these spikes are in this area here. And yeah. So this is, let me see. Okay, there are the interval counts. Okay.
Okay. I think then the thing to do is try to find a movie of a single moving diatom.
Okay.
And do a similar analysis, see if there's any qualitative differences.
Yeah.
Okay. Now, do you have a copy of Nick Heap's movies? Movie?
I don't know if I don't know if I do or not.
Okay, let me dig it out and send it to you.
Yeah, that would be good. Yeah. So those are just some examples. Yeah, I mean, if we had more data, I could do more with it. But yeah, I'm trying to figure out some of these dynamics within the time series. So we have a couple of different, from last week, we have a couple of different comparisons between different cells in the colony. So we have, so we have single cells and then relative movement of two cells. And so we have a fair amount of data for like one colony. So, you know, having other data would be maybe a bit confirmatory or, yeah. Good. So anyways, let's see. I'm going to move on to this thing I wanted to show on gene switches. So I've been talking to my other group about this thing called switching, and it's kind of an odd thing to maybe it doesn't make any sense to people, but so in a bunch of different natural systems, you see this effect called switching. And what that means is that you have this sort of relationship where you have a system and it switches between two states. So, you know, you can think of like, you know, when you're asleep versus when you're awake, that's kind of a crude switch. But there are other types of switches, like genetic switches, where you can switch between one mode and another, or, you know, in other systems where you have two or more modes that you switch between for some reason. It could be adaptive, it could be just stochastic, whatever. And so in this case, this is actually something the synthetic biology people are doing. This is called a genetic switch. So what they're doing is they're building these switches. And this is actually a simulation of this. So this is NetLogo. You run this, you can run this simulation in NetLogo, and you can generate like simulations of these genetic switches. So, yeah.
Bradley, comment.
Okay.
I ignored it, but just yesterday I ran across a paper suggesting that bacteria can have what they call a chaotic state.
Oh, okay.
And that the chaotic state is vulnerable to different kinds of antibiotics than normal states.
Yeah.
And since I'm not interested much in antibiotics, I ignored the paper. If I come across it again, I'll send it to you.
Okay, that sounds good.
Okay.
Yeah. So yeah, you can have like cells can have different states. Sometimes they can be chaotic. Sometimes they can be, some other state. And this might be, differentiation is essentially a switch too. So, but at the genetic level, you have these these gene circuits that you can build using synthetic biology techniques, and you can build these switches that allow you. In synthetic biology, people are mainly interested in, like, say, creating different biochemical or compounds or biochemicals using something like bacteria. So you have a bacterial genome. You knock in these cells that they design, and the cell might have a switch at its promoter region, where then it switches between two different states or more, and it can generate things if you give it like a stimulus, like some sort of food source or light or something like that. That switch can flip on and generate what you want. So this is the synthetic biology version of this switch. And so how it works in this simulation, I think I talked about LAC-Z promoters a few weeks ago, but basically they use this LAC-Z gene, which is something you find in bacteria. It's a lactase production gene, and there's a system of like operons that turn these genes on and off. So this gene will produce this enzyme, but you have to have the right promoter in place to do this. And so this is a very famous system, this LAC-Z system in bacteria, and they can manipulate this. So this is the genetic circuit that they propose. It's a LAC operator, which is this lactase gene with a promoter. They have this signal that tells it what to transcribe. It's a switch that tells it to transcribe one thing or another, to turn on and off and so forth. And then you have this agent-based model where you can see the thing being produced as you turn the switch on and off. And so this allows us to simulate this process. I think this is a nice little introduction, not just to like this concept, but also to this idea of like building a genetic circuit. Then there's this other Weiss Institute, which is a biological institute. I think they're out of Boston. They've done a lot of work with these kind of switches in actual biological organisms. So their gene off switches tool up synthetic biology. It's the title of this article. This is some of the institute's propaganda, as it were, because they're a press release, so they're very positive about it. But I wanted to point out that you have these different types of switches that people are working on, not just with LAC-Z, but all sorts of different interesting types of elements that they're working on. So you can program a repressor element, for example, that allows you to repress certain genes at certain times and turn them on at certain times. So this is good. This is useful for, maybe like medical diagnosis or environmental remediation, or even like making, biofabrication, where you want to make things on a circuit or at a very small scale. So they talk about this. They talk about ribocomputing devices, which are kind of the larger class of technology here. Ribocomputing devices can sense multiple biological RNA signals simultaneously and act as molecular logic boards. So these are, things that you can use. If you can string these switches together, you can build logic, a series of logic gates that you can do computations with. So this is a very different approach to computation than your digital computer, where you have, electrons that are really doing these, passing through these logic gates. You have, genes that are switched on and off, and they're producing something. So that's but it's still computation because it has that same logical structure to it. So any desirable regulatory element for synthetic biology would be a device that can do exactly the opposite of this thing here, which is only if a certain combination of input signals is present does the device produce a desired output protein. So in synthetic biology, we also want to do that, but we also want to shut down the expression of a protein in response to a stimulus when it is no longer wanted. So we want to have that kind of control. And so in this case, they're able to program, they created a library of 100 repressors. So you can choose from 100 repressors to build these logic gates. They've been able to implement 15 of these in parallel. And so you can have them all working in parallel to produce different things, to shut different things off. And so this is a paper in Nature Chemical Biology where they've published this. And then they've also designed something called a toehold switch, which is a different type of switch. This is where you have RNA strands that detect trigger RNAs. So these are RNAs that trigger these promoter regions with virtually arbitrary sequences to activate translation of a linked protein coding RNA sequence. So this is like where you can control the transcription of a gene, and this is, I think, allows for greater control than the purely transcriptional switch, which is where the promoter merely gets turned on and off. This is triggering the things that sort of trigger the promoter. So this is in a cell. This is important because this machinery is sensitive to the sequence of the RNA, and so if the RNA is the right sequence, it will trigger the promoter region. But if you can, like, there's a lot of RNA in the cell, so if you can trigger the right RNA and get it to do what you want, that's actually more precise. So this is I think that's all I'm going to say on that. I think it's an interesting area. Yeah.
Yeah. This goes well beyond than the standard gene deletion methods.
Oh yeah. So I mean, gene deletion is and people have worked out ways to sort of make that more and more precise. So with a lot of the stuff with CRISPR, and where they can just cut out a piece of DNA. But this is where you're controlling sort of the expression of genes. You can put genes in and build actual logic gates. So this means that you can, under certain conditions, you can express a gene the way you want it.
Yeah.
Yeah.
Okay.
So
Beginning of biorobotics.
Okay, yeah. So I'm gonna finish up with papers. Did we have anything else we wanted to mention, or? I know Susan has to leave in about 10 minutes, so I'll move on to some papers here.
Hello, did you want to talk to me, or?
Oh no, I just wanted to make sure that you didn't have anything else you wanted to mention before.
No, I need to say goodbye now and
Okay.
pack up and go.
Okay. Well, have a good week. Thanks for
Okay.
presenting.
Take care.
Take care.
Okay, thank you.
Bye. So I had a paper that let's see. This is the one that I didn't get last week because I misplaced it, but there's a new type of method here. We've talked about cellular automata in the group here, and there's someone from University of Manitoba. So this is called learning graph cellular automata, and this is we've talked about cellular automata in the group before as a way to model morphogenesis and different things like that with respect to development in biology. And what they're doing here is they're applying graph neural networks to cellular automata. And so let me go through this a little bit. So this isn't, I don't think this talks about graph neural networks, but I think this is, the idea is they're using these graphical structures, graph neural networks, and they're building these as sort of the back end of a cellular automata, which is this grid where you have cells that behave according to their neighbors. So the abstract reads, cellular automata are a classic computational model that exhibit rich dynamics emerging from local interactions of cells, arranging a regular lattice. So in this work, we focus on a generalized version of a typical CA called graph cellular automata. So this is where we have the graph part, the cellular automata part, in which the lattice structure is replaced by an arbitrary graph. So in this case, they're taking the lattice and they're replacing it with a graph. In particular, we extend previous work that used convolutional neural networks to learn the transition rule of conventional CA. And we use graph neural networks to learn a variety of transition rules for GCA. So they are using graph neural networks here. So in this previous work, they used a different type of neural network to learn transition rules. So basically, in cellular automata, you have these rules that they follow. I think we've talked about like Rule 120, which will give you something that looks like a seashell pattern, or different rules that will give you different patterns. And so they follow these rules. The rules based on their interactions will produce this macro pattern. In this case, what they're doing is they're taking a neural network to learn these rules, but they're replacing the conventional neural network with a graph neural network. So now they're learning a variety of transition rules for this type of cellular automata. So this paper, I think, just goes through how this is done. They present a general purpose architecture for learning a graph cellular automata. Then they show it can represent any arbitrary graph cellular automata with finite and discrete state space. So this graph neural network will learn enough rules and enough about the rule structure to reproduce anything that cellular automata might produce. So I don't know what Stephen Wolfram proposed, something like 200 and some rules for cellular automata. And what they're saying is they can reproduce most of those or all of those using just this graph neural network. Then we test our approach on 3 different tasks. So the first is learning a transition rule of a graph cellular automata of what they call a Voronoi tessellation, which is a type of tessellation of space that has continuous structure. They're partitions. So there's that kind of tessellation. It's imitating the behavior of a group of flocking agents. So this is where you're learning this transition rule on what is essentially a grid, and you're tessellating a space, and you're allowing the cells to evolve these global patterns. 2 is where you're imitating the behavior of a group of flocking agents. So this is, of course, like the Boyd simulation, or like something like a bird flock, or some other type of collective behavior. So they're just imitating that behavior. And then 3, learning a rule that converges to a desired target state. So this is where you might define a pattern, you might define the shape of an animal or something, and then the system will have to fill in that pattern somehow. It'll have to figure out the rules to get to that pattern. So this is actually, I think the third one is probably very close to the sort of, pattern formation and development that we see.
Hey, Bradley,
Yeah.
is this at all similar to Portuguese?
I don't think so. Well, I don't think so. I think Tom actually he does the cellular automata with different neighborhood sizes, and then he uses a neural network at the end to classify them. But this, I think, is actually where they're actually trying to learn the rules directly. I don't know if he's doing that step. I think he's just kind of classifying the outputs.
Yeah, I remember discussing that, but
Yeah.
I don't think it came to a resolution.
Yeah, but I think the point here is that they're trying to improve upon, people have been using neural networks for that purpose, to learn the rules themselves. But it's not easy to do, of course, because it's a huge state space, and you need to,
Yeah.
But so these graph neural networks apparently can do this, and other neural networks can't necessarily do this. So let's see if I can find some of the stuff down here. This is a nice paper because it gives you some, past work on this, where people are kind of building up to the this work. So a seminal work on the subject is Wolf and Hertz, who successfully trained a small neural network to imitate 1- and 2-dimensional binary cellular automata with chaotic or complex dynamics. Then if you also used evolutionary algorithms to identify neural network transition rules that would generate these patterns. So this is what they call morphogenesis. but this is, I don't know if that's, so they're just identifying here, they're just identifying these transition rules. So it's like, how do you get a rule that produces some sort of pattern that looks like it's, coherent. They also use the neat genetic algorithm, which is this neuroevolution topology. It's a little bit different type of genetic algorithm. It sort of moves in the direction of neural networks. And then compositional pattern producing NNs, which are the CPNNs, as well. So there's been past work on this. And then, people have used encoders, things like that. And then more recently, people have looked at neural cellular automata, which are these NCAs. And this is basically taking a convolutional neural network and using it to learn the transition rules of a two-dimensional cellular automata. This is interpreted as an image. And then they train the neural cellular automata to converge to a specific image. So you have this thing that you're defining, which is different than, of course, maybe morphogenesis, where maybe you don't have a target, you just have rules that give you the target consistently. But in this case, they're actually saying there's a target that you have to reach. And I've seen people do this before in different ways, like, they'll use a target as a training, as a training set, where they'll say, we want this pattern of a barbell. And so then the neural network will produce this pattern of a barbell. But that isn't necessarily morphogenesis. It's just kind of evolving this cellular automata to a target. So it's a very subtle difference, but it's important. Nevertheless, you get this global pattern that emerges that looks, very much like something you would find in a cellular automata, maybe even in morphogenesis. So this is actually here where they're able to train this cellular automata to form a rabbit. So they goes through like 20, I guess t equals 20, which is their time. They get a lot of error here over different steps, but they're able to get something that looks similar to a rabbit. They're able to form letters, GSP, in the same way. The error here is a lot less variable, so you can see that there's, and then, you have all these other things that they're trying to sort of attain, and these are patterns that they're seeking at the endpoint, but they use this to try to get to that. So this is an interesting paper. I think there's much more to be said on this topic. I wanted to go over this because we've talked a lot about neural networks and then graph neural networks. So I think this is in cellular automata. So I think this is a really interesting area. Yeah. And then let's see, maybe one more paper here. What is this one? Oh, this is a bunch of papers. We could talk about this. So this is the cell biology models of development. So this is a set of papers that have come out recently. This is on...
Bradley, I gotta go.
Okay. All right. Well, take care. All right.
Take care. Bye.
Take care. Bye. So this is a group of papers on cell biology models of development. This is looking at some of these developmental things in cell culture. So we have this, the mutational landscape of human somatic and germline cells. This article is, so the abstract reads, over the course of an individual's lifetime, normal human cells accumulate mutations. And so we know this from just studies that in your lifetime you accumulate mutations by interacting with your environment. But your germline, which is the thing that that your offspring will inherit, is protected from those sort of somatic mutations. Those cells are sequestered in a germline pretty early in development, and then they don't suffer those type of mutations. They have a different set of mutations, perhaps, but it's a little bit different. So over the course of an individual's lifetime, normal human cells accumulate mutations. Here we compare the mutational landscape in 29 cell types from the Soma and germline, using multiple samples from the same individuals. So they're looking at the germline, which is largely these undifferentiated cells, and you have the somatic cells, which are different cell types, like skin cells and bone cells and muscle cells and so forth. Two ubiquitous mutational signatures, SBS1 and SBS540, accounted for the majority of acquired mutations in most cell types, but their absolute and relative contributions vary substantially. SBS18, which potentially reflects oxidative damage, and several additional signatures attributed to exogenous and endogenous exposures, contributed mutations to subsets of cell types. The rate of mutation was lowest in spermatogonia, which is in the germline, the stem cells from which sperm are generated, okay, and from which most genetic variation in the human population is thought to originate. So this is a germline cell. It's experienced a very low rate of mutation. Now, germline cells will experience mutation, but they won't be environmental. They'll just be like random mutations. So you'll see very low rates of mutation in the germline. And so these stem cells from which sperm originate are protected by and large from mutation. And so this is where a lot of our genetic variation originates, but it's not deleterious. So this was due to low rates of ubiquitous mutational processes and may be partially attributable to a low rate of cell division in basal spermatogonia. So every time the cell divides, you get an opportunity for mutation, you get an opportunity for recombination, and so forth. And so low rates of cell division can also contribute to that. These results highlight similarities and differences in the maintenance of the germline and soma. So as I said, the germline is something that gets protected from a lot of environmental mutation. It's and this study is actually does a lot of this sort of, they kind of go over a lot of what's going on in these different cell types in terms of its mutational landscape. So they use next generation sequencing for this, and they align these next gen sequencing. So the next gen sequencing here is large scale transcriptional analysis and sequencing. So you can actually look at like the genes that are expressed in their frequencies of expression, but also the sequences that they come from. And so you can get all of these data, and it'll show mutations between cell lines. You can identify them. And so this just kind of shows the donor here, where they take, I guess they're harvesting from a dead body, or they're doing biopsies. And they basically, the worm autopsy and additional donors. So it's a very recently dead person, and they, when they do the autopsy, they can do a biopsy on the tissues. They can get these cells and look at the mutational signature. They can identify the cells via laser microscopy. Then they can do these samples. And these are some of the sort of the histology of these different cells. They can stand with antibodies, so they show some of the expression of some of these monoclonal and clonal and polyclonal antibodies in the histology here. So they're able to identify different mutations in the tissues. So as we were talking about before, we're talking about forces on tissues and their effects. These are mutational effects on tissues, so it's quite a bit different, but it's still, you can see a lot of variation across cell types. You can see that you have pancreas and kidney and prostate gland cells, stomach gland cells, all these different areas of the body, and you can see the sort of mutational signatures in these. And so yeah, mutation rates are interesting because you can calculate them. We don't, we kind of make like assumptions about the calculations, and we kind of try to figure out what the rates are, and of course the rates increase or decrease by exposure to environmental factors like sunshine or smoking or different things like that, radiation. But then, you can also have mutational rates that vary across cells. And I think before this paper, before the series of papers, people didn't really understand exactly what those look like across cell types. So this is another example here where you have the contribution of different mutations to different cell types. And you can see there's variation here. Skin is incurring a lot of mutations, I believe. Whereas in the germline, which is over here, you don't see very many mutations. So yeah, and then there are different mechanisms that underlie this mutation rate, differences in the mutation rate. There's also DNA repair. So when you have, say, like a skin cell that's exposed to the sun too long, you can have mutations in that cell, but then the DNA can also be repaired, so it's not something that is a long-term thing. So you get these, dynamics of mutations. Sometimes they drive a cell to a cancer pathway, sometimes they get repaired, and it depends on the cell type. So I'm going to go to this paper, which is this clonal dynamics in early human embryogenesis, inferred from somatic mutations. So now we're talking just about somatic mutations. And the abstract here reads, cellular dynamics and fate decision in early human embryogenesis remain largely unknown owing to the challenges of performing studies in human embryos. Here we explored whole genomes of 334 single-cell colonies and targeted deep sequences of 379 bulk tissues obtained from various anatomical locations of 7 recently deceased adult human donors. So again, they're using the recently deceased. They're doing the tissue biopsies, and they're getting cellular material for this study. Using somatic mutations as an intrinsic barcode, so they have this technique called barcoding where they can identify some of these mutations, and they can use it as a way to identify different cell types and things like that. We reconstructed early cellular phylogenies that demonstrate. So a cellular phylogeny is, I think we've talked about phylogenies before. They're these trees that show how different taxa diverge from one another historically. So in this case, we have cells that are developing, and they're able to show these phylogenies of cell types, how they diverge in terms of their mutational rate. So this is an interesting exercise. These cellular phylogenies demonstrated 5 things, actually. So the first is an endogenous mutational rate that is higher in the first cell division, which is the first cell division after it differentiates, but decreases to approximately 1 cell per cell division later in life. So this is where 1 per cell division. So this is where the mutational rate is high as it first is, that cell type is born, or that cell is born. And then as the cell divides, it gets fewer and fewer mutations. Interestingly, cells, of course, unless they're stem cells, they age as they divide. So they have this limit where if they make enough divisions, they undergo this apoptosis process and die. And so as they're getting closer to that apoptotic endpoint, they're actually getting fewer mutations in their genome. So this is an interesting sort of paradox, I guess. 2, universal unequal contribution of early cells to the embryo. There is a universal unequal contribution of early cells to the embryo proper, resulting from early cellular bottlenecks that stochastically set outside epiblast cells within the embryo. So this means that not all cell types are equally represented in the embryo. So this is just something that they're identifying from the mutational signature. Number 3, examples of varying degrees of early clonal imbalances between tissues on the left and right sides of the body. So there's this difference in terms of laterality. Your left side of your body and your right side of your body are supposed to be symmetrical, but sometimes they're asymmetrical. And this is one example of that. Also, different germ layers in specific anatomical parts and organs also exhibit this clonal imbalance. So this means that they're just asymmetries between the tissue types or mutational signatures in different tissues in different parts of the body, depending on where we're looking. 4, emergence of a few ancestral cells that will substantially contribute to adult cell pools in the blood and liver. So you have these cells that are just a few ancestral cells that go on to form the blood and liver, proliferate, and form these different things in the body. And then 5, presence of mitochondrial DNA heteroplasmy in a fertilized egg. So this is where you get multiple mitochondrial DNA in the fertilized egg. So these are all things they can find from these somatic mutations. Our approach also provides insight into the age-related mutational processes and loss of sex chromosomes in normal somatic cells. In sum, the study provides a foundation for future studies to complete what they call cellular phylogenies in human embryogenesis. So this is something that they go back, they look at like embryogenesis from the adult cells in the body. They trace these cells back to their embryonic origins, and they can make these inferences through the mutational signatures, and they can find out all these things. It's kind of a really amazing paper. I don't know how, I'm not going to go through the entirety of it, but I mean, you can read it yourself. But
yeah. Like coming to even SNPs, I think, like, is it kind of something similar? Because mutations has happened both ways, like germline and somatic one. So maybe these differences kind of develop into those SNPs, right? Maybe.
Yeah.
That type of gene variation across people.
Yeah. Yeah.
I also had some other ideas, that I wanted to bounce off, because I'm still, kind of new to all this, but still, like, I can grasp some things, and, some things are kind of, like, still in the dark, so I'll have to, go through a bit more terms. But, like Susan's PPT, which was discussing viscous elastic properties of tissues, and, the kind of applications that we can use them for, like, building tensor models for, let's say, a physical medium, where we can, calculate heat stresses or load stresses and all that. So that is kind of easy, because there are a lot of libraries for that, Python libraries or MATLAB. We can do it on MATLAB as well. So when it comes to potential applications, like there's this idea, like, where we have, piezoelectric material, because essentially what we'll be doing is we'll be mapping out the stress and strain relationship of this tissue, which is in the form of a tensor model. So maybe, data in terms of, like, the amount of energy that can be, transferred, or the work that can be done to maybe power, wireless implantable medical devices. Because this is, again, a field of, papers that I was going through. So the problem in this field is a lot of devices that are used here, they have to be powered, wirelessly from magnetic fields or electromagnetic induction and all that. So the challenge right now here is, finding viable sites inside the human body. Like some people were, suggesting the cardiovascular system for propelling maybe, devices that flow through your bloodstream. So maybe something along these lines, but a bit more stationary, like maybe it has a target site where it is close to your heart, like for, let's say, pacemakers or your muscles. So it will kind of, like, given this kind of data, we can kind of carry out a test for viable sites where we can, put these kind of devices.
Yeah.
Maybe, like, just an idea that's there.
Yeah.
So at least coming up with the model for TensorFlow plot, like, state-of-the-art things are there. Like, I think it won't take a lot of time. And there was another thing I had, like, I was not able to find, accurate data when it came to axolotl and zebrafish model. Like, for doing carrying out the 4D model thing, right?
Yeah.
The data that is there, like, if you could, give me some references or resources for that.
Yeah.
That'd be really helpful, because so that I can fiddle around with the data, right?
Right. I'll take a look. I know we have some data on. It's kind of hard to find data. We have some zebrafish data that are like cell tracking data, and then we have some raw axolotl data, which is just it's actually a three-dimensional. Well, it's two-dimensional, but it's three-dimensional, and it could be four-dimensional if you can extract the data points from the images. So that's a stumbling block. But there is data on that. It would have to be, you'd have to maybe play some tricks a little bit with the modeling of it. But
yeah, I mean that's what I wanted to fiddle around, so that like with the time sequencing and all that, how to represent it in slices or something.
Yeah.
So
I'll send you
It-
along
that
some
will
of
be
the
helpful.
data. Yeah.
okay. And this thing, I don't think it will take a lot of time, but that again depends on the kind of viscous elastic material Susan was talking about. Maybe because generating a tensile model with physical medium doesn't take that much time, but tissues, I guess, will be complicated here
Yeah.
because we're dealing with continuum mechanics, so
Yeah.
that will be there. And yeah, like gene sequencing, protein sequencing, and all these things, these are still like when it comes to graph cell automata and those somatic mutations and germline mutations.
Mm-hmm.
It's still we'll have to go through stuff,
Yeah.
before kind of grasp or get at least some part of the bigger picture in your head.
Yeah.
Because I can get understand some portions of it, but overall I think it's still fiddling with things in the dark, right? But yeah, like it's kind of interesting,
Well, yeah. I mean, this is just like it's one of those things where you just have to like see it enough and let it wash over you, and you can kind of catch on to what they're doing. And so
Yeah.
good.
Yeah.
Well,
Okay.
yeah. If that's I don't think I have anything else for today. So thank you for attending, Karan, and I'll send you some data. I'm glad to hear that you're interested in some things that are, asking about different data sets and things like that. So
Yeah, I mean, it'll always be helpful because to middle around with those things, you'll have to see what will properties I'll have to consider, with that thing, and especially when generating that 4D model thing, it becomes kind of heavy on the computer as well because processing a 4D image across time, depending on the again complexity of the model, those factors will also have to be considered.
right.
But yeah, definitely like all help will be appreciated because finding data sets right now, especially with regards to axolotl data or zebrafish, in going through the developmental stages from an embryo to the adult state. And also with regards to complexity, I think zebrafish has lesser number of stages, right, compared to an axolotl. Like it's much more easier to study.
Well, zebrafish has a fair number of stages, but I don't know compared to axolotl what might be because zebrafish has a number of different stages that are kind of like they're just the way they've classified them is. I think it's at least 10. I have a list of the different stages I can send you along with the data. So it's, but it's it's less maybe less complex from the standpoint of like actual morphogenesis where it's forming new things. So
that probably was why I was considering zebrafish is maybe has lesser development stages than axolotl.
Yeah.
But yeah, definitely worth getting into.
Yeah.
So yeah. I'll let it go to it.
All right. Okay. Well, thanks for attending, and thank you to Dick and Susan for their participation. And yeah, let's talk more on Slack about this.
Okay.
Okay.
Yeah, definitely.
All right.
Thank you, Bradley.
Have a good week.
Yeah.
Bye.
Yeah.